Hi, my name is Grace McNally, and my paper presentation will be on signatures of T cell dysfunction and exclusion to predict cancer immunotherapy response. So I will first start out in this presentation by talking about the context of the research. Then I will move on to the major findings of this paper, then my own personal scientific critique, and then the impact this paper had. So, background and significance. I will first start by discussing immune checkpoint blockade therapies, or ICB therapy. ICB therapy is a type of immunotherapy to treat various types of cancers. The therapy can be very effective, offering long-term and durable results, even in advanced stage metastatic cancers. It enhances the immune system's ability to detect and destroy tumors by targeting immune checkpoint proteins. These proteins normally act as regulators to prevent excessive immune responses, but can be exploited by cancer cells to evade immune attacks. Although this therapy may be beneficial, not every patient responds to this treatment. This is why the authors of the paper developed a computational model called TIDE. TIDE, or Tumor Immune Dysfunction and Exclusion, is a computational framework developed to test whether or not a patient will respond to ICB therapy by using the tumor's transcriptome, which refers to its complete set of RNA transcripts. This model has utilized 189 publicly available human cancer studies to compile a large set of gene signatures to develop this framework. The authors previously identified two distinct mechanisms of immune evasion to discover what underlying functions may result in ICB resistance. The first mechanism is T cell dysfunction. In a healthy system, immune cells called T cells play a key role in identifying and destroying cells that may be harmful to the host. In a cancer patient, several factors may impair proper T cell function. T cells may be able to infiltrate tumors, but they are non-functional and lack the ability to kill and fight off cancer cells, allowing for the tumor to evade a proper immune response. The other mechanism the authors looked at was T cell exclusion. This is when a tumor blocks T cells from entering using immunosuppressive factors. This inhibits the ability of the immune system to kill cancer cells. Currently, the known factors that affect ICB success listed do not provide valid response and resistance predictions to therapy. ICB clinical trials use small numbers of gene expression samples from pre-treatment patients, which is not significant in identifying biomarkers for prognosis either. Although ICB may offer some patients long-term and beneficial results compared to conventional cancer therapies, patient response to ICB therapies may be unpredictable. Only 1/3 of patients respond to ICB therapy. There are currently no known biomarkers that would result in an accurate prediction for patient response. The authors identified the need for an accurate way to identify resistance and response regulators for ICB therapy across large populations of cancer patients. So now I will move on to the major findings of this paper. So the authors first focused on identifying the gene signatures of tumors that evade immune response using T cell dysfunction. They compiled data from treatment-naive metastatic melanoma tumors, along with the patient survival data to pinpoint the interactions of T cells and a known regulator of T cell dysfunction, TGFB1. First, the authors estimated the cytotoxic T cell lymphocyte levels, or CTL, by averaging the expression of the genes associated with CTL activity shown here. Um, uh, Using a Kaplan-Meier plot, the authors plotted the survival fraction of patients with either high or low CTL levels with different TGFB1 levels shown in figure A here. The authors calculated the survival outcome based on the interaction between CTL and TGFB1 using the Cox proportional hazard model shown in figure B. They were able to confirm the known antagonistic interaction of the two variables through this model. After obtaining results that agreed with the known outcomes, the authors were able to use TIDE to test how other candidate genes interact with CTL levels to affect survival outcome using the Cox pH model. TIDE was used on hundreds of publicly available cancer data sets to predict statistically significant interactions between genes and CTL. Although some genes identified are usually co-expressed with immune suppressive genes, some genes were known to regulate T-cell mediated tumor immunity. The authors were able to identify these genes of interest, and those are marked with a star here in Figure C. So, the next major finding focused on identifying and modeling tumors that utilize the mechanism of T-cell exclusion. The authors looked at three types of cancers known to exclude T cells as an immune evasion mechanism: cancer-associated fibroblasts, myeloid-derived suppressor cells, and the M2 subtype of tumor-associated macrophages. The gene expression signature of T cell exclusion on the x-axis for each type of cancer was plotted against their corresponding CTL levels on the y-axis. They found a strong negative correlation between T cell levels and T cell exclusion scores across all cancers. This result agrees with the thought that tumors that use T cell exclusion will have less T cell infiltration, and this model allows TIDE to be able to identify T cell exclusion in tumors. So, in order to have TIDE identify the mechanism a tumor is using for immune evasion and differentiate between the two, the authors looked at the gene signatures of both T cell dysfunction and T cell exclusion at the same time. The authors computed and plotted the Pearson correlation between the TIDE T cell dysfunction signature, shown on the y-axis, and the TIDE T cell exclusion signature, shown on the x-axis. The results shown in this figure D were consistent with known high CTL levels in kidney renal cell carcinoma, shown in the upper right corner, or upper left corner, excuse me, and low CTL levels in lung squamous carcinoma, shown in the bottom right corner. This result agrees with these results suggest that kidney renal cell carcinoma utilizes T cell dysfunction as an immune evasion mechanism, while lung squamous carcinoma utilizes T cell exclusion. Using this, TIDE is able to identify which mechanism is being used through immunosuppressive signatures. So, after modeling the gene signatures of both T cell dysfunction and T cell exclusion, and modeling the correlation between the two, the expression signatures were integrated to determine ICB response. So, tumors with high CTL levels, TIDE identifies as utilizing T cell dysfunction, and tumors with high T cell dysfunction scores are predicted to be non-responders to ICB therapy. Tumors with low CTL levels, TIDE identifies as utilizing T cell exclusion, with low T cell exclusion scores predicted to be responders. Tumor expression and patient outcome data was collected on melanoma tumors. Tumors were divided into either high or low CTL levels. The TIDE prediction scores were obtained from the previous Pearson correlation between T cell dysfunction and T cell exclusion. The waterfall plot shown in figures A through C show TIDE prediction scores across each tumor, with red indicating the patient responded to therapy and blue indicating the patient did not respond to therapy. To examine the results of these TIDE predictions, the authors used a receiver operating characteristic curve, or ROC, to identify the false positive rate against the true positive rate at multiple TIDE prediction scores. So this is shown in figures D through F. The black curve shows the performance of TIDE, and the purple, green, and red curves show the performance of predictions of currently used biomarkers to determine ICB response. Figures G through I show the area under the ROC curve, which is used to evaluate performance. As you can see, TIDE prediction model ranks significantly higher than all other biomarkers, which with the majority of the values at or above 0.8, the clinical standard. This shows that TIDE is a better predictor of patient ICB response compared to any other currently used signatures. So, for the final finding, the authors looked at genes of interest identified previously by TIDE as potential ICB resistance regulators. The authors first started by comparing the log-fold change of a gene expression between ICB-resistant tumors and the parental melanoma tumor. Each data point on Figure A is an independent gene of interest with a high TIDE T-cell dysfunction score. It was found that SERPINB9, shown in the top right corner, to be the most upregulated gene. This gene encodes for a protein expressed by lymphocytes that binds with cytotoxic protease to protect cells from apoptosis. Cancer cells expressing this protein may use it to prevent T-cell mediated killing during ICB therapy. In order to validate the function of this gene in cancer cells, an experiment was performed to knock out the SERPINB9 gene using CRISPR-Cas9 in a melanoma cell line. Once the protein expression levels were undetectable in the cells were then co-cultured with cytotoxic T cells. They found that cancer cells with this knockout were more susceptible to T-cell mediated killing compared to that of the control. To further validate this hypothesis, the authors engineered a melanoma cells to overexpress SERPINB9. These cells were maintained until they were expressing high protein levels, and then they were co-cultured with cytotoxic T cells. They found that cancer cells that overexpressed SERPINB9 were more resistant to T-cell mediated killing compared to the control. These findings experimentally validated that SERPINB9 regulates T-cell mediated killing, a process which is critical to effective ICB response. This further validated TIDE's ability to locate and identify novel genes that may play a role in ICB resistance. So, over the course of this paper, the authors showed the development of a novel computational framework that incorporates two distinct mechanisms of immune evasion to identify ICB response. TIDE was able to identify gene signatures of tumors that use T-cell dysfunction and gene signatures of tumors that use T-cell exclusion and differentiate between the two. The model developed is able to assign a TIDE score that is more accurately predicts a patient's response to ICB therapy compared to traditionally used biomarkers using pre-treatment tumor genomes. The authors also demonstrated TIDE's ability to identify novel regulators to ICB resistance for therapeutic knockout in order to improve ICB response, shown through the discovery of SERPINB9 as a regulator. Okay, so now I will be moving on to my critique of this paper. So my first critique of this paper is the incomplete reporting. The TIDE framework utilizes publicly available transcriptomic data sets, which allow for large population size of over 33,000 samples, and mainly focus on modeling melanoma. This paper, however, does not include any demographic data on any of the sample sets besides cancer type. Representation in medical studies typically have disproportionately high levels of white men and women. In a study called The Ongoing Racial Disparities in Melanoma, an analysis of the Surveillance, Epidemiology, and End Results database done in 2020, the authors found out that out of 398,034 melanoma patients, 95.4% were non-Hispanic white. Although majority of patients were white, minorities regularly suffer worse melanoma-specific survival. This racial disparity and of sample size and survival outcome should not be overlooked when creating a computational model. This may lead to TIDE performing poorly in underrepresented groups, resulting in worse outcomes. Future sampling through TIDE should incorporate demographic data and recognize inherent biases and imbalances. This may lead to improved prognostic outcomes for underrepresented groups. So my next critique on this paper is the simplification of immune exclusion. CTL levels are a major tide identifier in ICB resistance. The authors in this paper binarily classify CTL levels as either high or low. This gross oversimplification does not capture microenvironment interactions in tumor cells, T cell trafficking, or patient-specific variations. This may result in overfitting for data sets with different immune profiles that do not fit into a binary model. Overfitting resulting in inaccurate prognostic decisions may be detrimental to a patient's well-being. Rather than using a binary model, I propose the authors use a generalized linear mixed model, which may be more suitable for a suitable framework that would treat CTL levels as a continuous variable. This would account for fixed variables like tumor type or gene expression, as well as random effects like cohort differences and patient-specific variables. Would allow for continuous CTL modeling for tumors that may utilize varying levels of both T cell dysfunction and T cell exclusion, while also accounting for variability across samples and patients. This avoids inherent oversimplification that occurs in binary models and allows for more robust predictions to capture complex immune exclusion dynamics. So when looking at TIDE's ability to predict ICB response, the authors used a ROC test on cohorts tested with different treatments. The three cohorts consist of 25 samples, 42 samples, and 33 samples. The sample sizes are insufficient in offering a robust ROC analysis. To perform this test accurately, it is typically recommended to use 100 to 150 samples. With such a small sample size, the ability to detect meaningful changes for the area under the curve decreases significantly. This is critical due to the authors basing TIDE superiority over traditional biomarkers based on the area under the curve values. There is also an increased risk of bias, where the TIDE model may have performed well in a small sample size, but fails to generalize a larger population. In a future study, a ROC test performed on cohorts of 100 or larger may offer more comprehensive and accurate results. So now I will be moving on to the impact this paper has had on the scientific and medical community. So this paper has introduced one of the first frameworks that uses multi-gene signatures to predict ICB response more accurately than any other current single biomarkers. The framework serves as a cornerstone for the development and refinement of more multi-factor predictive models for a wide range of cancers. TIDE may be beneficial in clinical settings, allowing patient-specific immunotherapy treatments. By integrating patient tumor gene expressions, patients that are predicted to have poor responses to ICB may seek alternate treatments and avoid unnecessary complications, while patients predicted to have beneficial responses are able to have more targeted use of treatments. This paper has also identified a new regulator of ICB resistance, which may be explored as a potential therapeutic target. Although there is no current inhibitor available, the Pfizer OASIS database stipulates that targeting this protein for therapeutics may actually be possible. This discovery has opened new research avenues for developing therapies to enhance the effectiveness of immunotherapies for the next generation of cancer treatments. Thank you very much for listening to my presentation, and please feel free to ask any questions. Thank you.